Hello guys, I'm Vineet and I welcome you all to the molecular biology series chapter number 1. Today I'll be discussing about the nucleotides, the structure, properties and functions of the nucleotides. So if we talk about the nucleic acids, the major hereditary materials uh, which are present in the nature is the DNA, and some viruses also contain the RNA as the genetic material. Okay, so these nucleic acids, DNA and RNA, are made up of the nucleotides. Okay, so these nucleotides actually stacked upon each other. So if this is the simple nucleotide, so it stacks upon one nucleotide. Okay, and one more nucleotide stacks upon another. Okay, and these are linked by a special type of bond known as the phosphodiester bond, like this. Okay, and becomes a long biopolymer chain. Okay, so one strand of the DNA, and if the same strand combines with another strand like this, which also contain the phosphodiester linkage. Okay, if this one biopolymer strand uh, combines with the second biopolymer strand of the nucleotides, it combines to form the DNA. Okay, so this will be the double helical structure of the DNA. Okay, and these two chains of DNA are linked by the hydrogen bonds. Okay, like this. Okay, so this is about the nucleic acids. And nucleic acids are the anionic biopolymers. That means they are the negatively charged compounds in the cell. So the negative charge on the DNA is mainly due to the presence of the phosphoric acids. Okay, so these nucleotides which are present in the nucleic acids give the negative charge to this entire biopolymer. Okay, and these nucleotides which make up the nucleic acids in turn made up of three components. So one is the nitrogenous base, and one is 5 carbon sugar, and one is phosphate group. Okay, and this phosphate group is responsible for the uh, giving negative charge to this DNA or RNA. Okay, so this is about the basic information of the nucleotides. Okay, so if we talk about the nitrogenous bases, which are the major components in formation of nucleotides and nucleic acids, not only serving as the major component for the building up of nucleic acid, they also take part in structural component for formation of the coenzymes. Okay, so the nitrogenous bases are also involved in the formation of the coenzymes, and there are a lot of many coenzymes in the biochemistry, but major electron donors or hydrogen transfer coenzymes are FAD and NAD. Okay, so these FAD and NAD transfer the electrons or the hydrogens, okay, to the compounds, and these NAD and FAD contains nitrogenous bases. Okay, while NAD contains two nitrogenous bases and FAD contains the NAD, it contains the two nitrogenous bases, while FAD contains only one nitrogenous base. Okay, so if we talk about the structure of this NAD, so this NAD primarily it contains one complete nitrogenous base, nucleotide. Okay, so in this nucleotide we have one nitrogenous base, one pentose sugar, and one phosphoric acid group over here. Okay, so this is the purine, complete purine. So this purine is actually an adenine molecule, and above the purine there is another molecule attached to the phosphate group of this purine. So that is called as the PRPP, 5-phosphoribosal pyrophosphate. So the yellow one which you are seeing in the image is the PRPP, and this PRPP is again linked to this pyridine link, which is also a nitrogenous base, and this pyridine comes from the tryptophan or nicotinamide. Okay, so whole together forms the nicotinamide dinucleotide. Okay, so the same structure goes for the FAD also, but FAD contains only one nucleotide, one nitrogenous base actually. Okay, so this is the major difference between FAD and NAD, and they also involved in the electron transfer. And the coenzyme which is like the vitamin which is involved in the formation of this NAD is niacin, or you can also call as a nicotinamide or nicotinic acid, and the vitamin which is involved in the formation of this FAD is riboflavin. Okay, so these two are the vitamins which are involved in the formation of the coenzymes. Okay, so nitrogenous bases not only serving as the major components for the formation of the nucleic acids, they also take part in formation of the coenzymes. Okay, so that is combining with the certain type of vitamins and forming a coenzyme. Okay, and if we talk about the properties of these nitrogenous bases, they are heterocyclic majorly, and they are planar and aromatic, and they are water insoluble. Okay, so heterocyclic is containing a different types of rings, and they are planar and they are aromatic because they contain the uh, benzene ring. Okay, and they also have the 2n+2 pi electrons in them, and they are water insoluble. Okay, so this is about the basic introduction about the nitrogenous bases. So if we talk about how do we extract these nitrogenous bases? Okay, the major question would be, how do we extract these nitrogenous bases from the DNA? So if you look at this DNA or the major nucleic acids inside any cell, bacterial cell, or in mammal cell or any vertebrate cell, these nucleic acids are present in combined form. Okay, so these nucleic acids combines with a certain type of positively charged proteins. So that might be the histones, so some other kind of protamines, or coenzymes contains, a lot of proteins are there which contains a lot of positively charged amino acids or basic amino acids. So these nucleic acids combine with those kind of proteins. Okay, if you want to simply extract the nitrogenous bases, first we have to separate these nucleoproteins into nucleic acids and the proteins. Okay, for that we have to add the proteases enzyme. Proteases cleave the protein from the nucleoproteins and separate the nucleic acids and the proteins. Then you get the nucleic acids, and to the nucleic acids we need to add the special enzyme known as the nuclease. Okay, so this nuclease, it is of two types, ribonuclease or the deoxyribonuclease, which breaks down the nucleic acid, okay, long biopolymer chain into simple molecules. Okay, simple monomers. Okay, so when it hydrolyzes the phosphodiester bond that is present between the nitrogenous bases in the DNA, so these nitrogenous nucleotides will get separated out. Okay, and the phosphodiester bond, because of the phosphodiester bond is broken off, these nucleotides will get separated out, and you will get the individual nucleotides in the mixture. Okay, and remember, you are carrying out this entire process in aqueous ammonia at 115 degrees Celsius temperature. Or if you don't have these conditions, you can also use the barium hydroxide solution. Okay, and you can also get nucleosides directly from the nucleic acid by when you are if you are doing this reaction in the magnesium oxide solution. Okay, I'll tell you this. So when this nucleus breaks down the phosphodiester linkage that is present between the two nucleotides, all the nucleotides get separated, and individual nucleotides are formed. Okay, to the formed nucleotides, then you have to add the nucleotidases. Okay, mononucleotidases or the phosphatases, which cleaves the phosphoester bond that is present between the sugar and phosphoric acid. Okay, so when you do this, the phosphate group gets separated out from the nucleotide, and you only have a nitrogenous base along with the sugar molecule. Okay, and to that, so nitrogenous base with sugar molecule, you call the structure as the nucleoside, and to that structure, if you add the nucleosidases, then you will get the sugar molecule as well as the nitrogenous bases. Okay, so these nitrogen bases are of two types, purines and pyrimidines. Purines are double ring compound, heterocyclic double ring compound, and pyrimidines are single ring compound. Okay, the major difference between the purines and pyrimidines is they contain purines contain an extra imidazole ring, while pyrimidines doesn't contain any imidazole ring. Okay, so this is how you extract the nitrogenous bases from the nucleoproteins. Okay, that the raw form of nucleic acid which is available in your nucleus. Okay, so as I said, you can also extract the nucleosides directly from the nucleic acid when you are doing that in a MgO solution. Okay, what happens is the magnesium ion, it's a divalent cation. It forms a interaction with this phosphate groups. Okay, and the charge get neutralized, and you can easily get the nucleosides. Okay, so that's a different way. And if we talk about the nitrogenous bases, the major nitrogenous bases which we have in our nature are 5. Okay, so adenine, guanine, uracil, thymine, and cytosine. And uracil is not present in the DNA while it is present in the RNA. So this is the major difference between the DNA and RNA. One major difference. Okay, and these adenine and guanine are considered as the purines, while uracil, thymine, and cytosine are considered as the pyrimidines. Okay, and purines contains the one extra imidazole ring. This ring is called as the imidazole ring. Okay, and this is called as a pyridine ring or a normal benzene ring. Okay, so the bigger ring. And if we talk about the chemical names of these purines and pyrimidines, okay, so the chemical names are dependent on the numberings. Okay, the type, different type of number which the atom gets. Okay, so the and the subgroup gets. Okay, so depend on the numbering, the chemical names also get differs in the purines and the pyrimidines. Okay, so if we talk about the numbering system in purines, so it goes in the anti-clockwise direction like this, while in the pyrimidines it goes in the clockwise direction. So this is the most important thing you have to remember for the entrance exams or the college level exams. Okay, so the numbering in purine is given in the anti-clockwise direction and pyrimidine is in the clockwise direction. Okay, and the name for the adenine, it will be 6 amino purine, because we have a amine subgroup over the 6th carbon atom, so the name will be written as 6 amino purine. Okay, and the name for the guanine will be 2 amino 6 oxopurine. Okay, because it has a oxygen group on the 6th carbon and amino group on the 2nd carbon, so the name will be 2 amino 6 oxopurine. And the same way the pyrimidine chemical names also given. Okay, so but in the clockwise direction. If we look at this uracil structure, the name for the uracil structure will be 2,4 dioxopyrimidine. Okay, 2,4 dioxo, and the name for the thymine will be 2,4 dioxo 5 methyl thymine. And the name for the cytosine will be 2-oxo 4-amino cytosine. Sorry, not thymine, it's pyrimidine. And here it will be okay, so 4-amino 2-oxo pyrimidine. Okay, so these are the chemical names of the nitrogenous bases, and these nitrogenous bases are majorly present in the nucleic acids. Okay, uh, purine contains adenine and guanine, and pyrimidine contains the uracil, thymine, and cytosine. The name for the adenine is 6-amino purine, while guanine is the 2-amino 6-oxopurine, and the name for the uracil, which is a pyrimidine, is the 2,4 dioxopyrimidine, and the name for the thymine is 2,4 dioxo 5-methyl pyrimidine. The name for the cytosine is 4-amino 2-oxopyrimidine. Okay, so if you look at the cytosine structure and the uracil structure, you know, there will be a lot of interconversion of the bases happening in the nucleic acids. Okay, so that will be the uracil getting converted into cytosine in unusual times. Okay, so the only thing that is different in uracil and the cytosine is this amino group. Okay, so if you remove this oxygen group, this oxo group, and if you add an amino group from the glutamine amino acid, okay, which is amino group donor, that will get converted into the cytosine. Okay, and the same reversal pathway will also happens. Okay, so this type of interconversion majorly happens in the DNA, and also this conversion of this uracil to thymine or the cytosine to thymine if there is any methyl group donor. Okay, so generally uracil gets converted into the thymine, or thymine also get converted into uracil, if there is any methyl group donor. Okay, so this kind of interconversion happens in the nucleic acids. Okay, so when we talk about the gene regulation, I'll discuss about these topics. Okay, so just remember nitrogen bases are of two types, purines and pyrimidines, and purines contains, purines are the double ring compounds, while pyrimidines are the single ring compounds. Okay, and uracil is present only in the DNA, while thymine is present in the uracil is present in the RNA, while thymine is present in the DNA. Okay, so this is the major difference between the DNA and RNA. Okay, And apart from the normal nitrogenous bases in the nature, we also have the unusual bases. Okay, unusual bases in the sense, which are the modified bases. Okay, the actual bases are modified into certain different types of things. Okay, so these modified bases, a modified form. So this modified bases has different functions. Okay, in the nature. Okay, like uh, the mRNA. So when this mRNA is fully processed and comes out from the nucleus for the translation process, like for the protein synthesis process, this mRNA contains a 5 prime cap, 7 methyl guanosine triphosphate. Okay, so this is a this compound or modified base serves as a cap for this mRNA molecule. Okay, cap like bottle cap. Okay, so this 7 methyl guanosine triphosphate, it protects from the 5 prime exonucleases in the cytosol. Okay, so whenever the mRNA comes from the nucleus as it is, there are a lot of 5 prime exonucleases which breaks the mRNA, or which degrades the mRNA molecule. Okay, in order to overcome the degradation process, so we add a 7 methyl guanosine triphosphate cap to the mRNA, so that this cap protects the mRNA molecule and also helps in the transportation process. Okay, so there are a lot of unusual bases in the nature. So first one what we have is a 5-hydroxymethyl cytosine that is in the bacteriophage, and second what we have is the N6-methyladenine or N7-methylguanine. Okay, so N7-methylguanine is adding a methyl group on the seventh nitrogen atom in guanine. Okay, so that is the major component in the formation of cap for this mRNA. Okay, so capping in the capping process. Okay, and we also have N6 dimethyladenine, uh, putting two methyl groups at the sixth nitrogen atom in the adenine molecule. Okay, so that becomes a N6 dimethyladenine. And apart from that, we also have a 5-methyl cytosine, just like what we have seen in the case of thymine. Okay, we also have the 5-methyl cytosine. Okay, the modified base of the cytosine, which looks like thymine. Okay, so this is the 5-methyl cytosine. Okay, and uracil modified at the N3 position by attachment of a propyl group has also been detected in bacteria, but this kind of unusual base doesn't have any kind of functions in the uh, bacteria. Like the functions are not clear, or are yet to be understand. Okay, so this is about the unusual bases in the mammals. Okay, so if we talk about the plants, there are also a lot of unusual bases like the caffeine, theobromine, and theophylline. These are the modified forms of nitrogenous bases. So the chemical name of this theophylline is 1,3-dimethylxanthine, which is found in the tea. And theobromine is 3,7-dimethylxanthine, and caffeine contains 1,3,7-trimethylxanthine. Okay, so xanthine is a nitrogenous base. So to this xanthine, you have to add some kind of extra methyl groups to it. So that depends on the different numbers or different carbon atoms or different nitrogen atoms. Okay, so that becomes a modified base. Okay, so theobromine, theophylline, theobromine, and caffeine, they differ only in the amount of methyl groups. Okay, theophylline and theobromine differs in uh, the position of the methyl group, that is 1 comma 3 and 3 comma 7, and theobromine and caffeine differs in the amount of methyl group. Okay, like only one methyl group is extra in the caffeine. Okay, so these are the modified nitrogenous bases which are present in the plants, and above we have the modified bases or the unusual bases which are present in the mammals. Okay, apart from this we also have lot of unusual bases present plants and animals. Okay, but these are the major ones. Okay, and if we talk about the nitrogenous bases, one major property of the nitrogenous bases is to exhibit tautomerism. Okay, tautomerism is interconversion of the a keto group to enol form. Okay, so this is called as a tautomerism, and this is one major property of this nitrogenous bases, and they exhibit it. Okay, so the base in the nitrogenous base which contains the keto group, we call that as the lactam form, and if the same keto group gets interconverted to enol form, so we call that form as the lactine form. Okay, so mostly the guanine, cytosine, thymine, and uracil exhibit this kind of tautomerism, but while adenine doesn't exhibit any kind of tautomerism because it doesn't contain any kind of keto group, it only contains amino group. It exhibits the amino form, the kind of tautomerism. Okay, but this tautomerism which involves the keto group and enol form is not exhibited by the adenine. Okay, so this is the major important point for the entrance level examinations. Okay, and this property uh, uh, will also be exhibited by different types of amino acid which has aromatic rings on and keto groups over them. Okay, so this is known as the tautomerism. Okay, so conversion of the keto form to enol form. And one major property of this nitrogen bases is to exhibit the tautomerism. Okay, and nitrogen bases, now if they want to form a next level structure, they have to combine with the sugars. Okay, so when these nitrogen bases combine with the sugars, they form the nucleosides. So these are the first level structures in formation of the nucleic acids. These nucleosides, which contain the pentose sugar, this pentose sugar is linked to the nitrogenous bases by glycosidic bond. Okay, so this glycosidic bond is N-linked glycosidic bond because nitrogen atom is involved in formation of the glycosidic bond between the nitrogenous bases and the pentose sugar. Okay, and the sugars which are present in the DNA and RNA, both nucleic acids, is different. Okay, so the DNA contains the deoxyribose sugar while the RNA contains the ribose sugar. Okay, so the only difference is DNA doesn't have any kind of oxygen atom at the carbon 2 position, while the RNA, the sugar which is present in the RNA, contains the oxygen at the carbon 2 position. Okay, so since we don't have this oxygen at the carbon 2, we are calling that as a 2-deoxo. Deoxo in the sense, we are removing an oxygen group. Okay, 2-deoxyribose sugar, this is present in the DNA, while the ribose sugar, normal pentose sugar, is present in the RNA. Okay, and these riboses mainly comes from the HMP pathway. So that is a hexose monophosphate pathway. Okay, so this for the synthesis of this the ribose sugars, we have one separate pathway called HMP pathway. So they come from the HMP pathway. Okay, and the purines and pyrimidines involves different nitrogen atoms to link this ribose sugars. Okay, like in purines, 9th carbon atom is involved in formation of the glycosidic bond with the sugars. Okay, like this. Okay, and in the pyrimidines, first nitrogen atom is involved in the formation of the glycosidic bond with the okay, so in pyrimidines, the N nitrogen atom 1. Okay, so first position nitrogen atom is involved in the formation of the glycosidic bond with the sugar molecule. Okay, so this is also a major difference. As I have said you, the pentoses are bound to the nitrogenous bases by beta and glycosidic bonds, and the nitrogen 9 of a purine ring binds with the carbon 1 of a pentose sugar to form the covalent bond. Yeah, this glycosidic bond is a covalent bond, and the purine to form the purine nucleoside. And in case of pyrimidine nucleosides, the nitrogen 1 will be involved in the formation of the glycosidic bond. Okay, so this is about the sugars which are found in the nucleic acids. Okay, and there is also one major property of the sugars in this nucleic acid, to exhibit the antisyne conformation and the puckering of these sugars. Okay, so I'll be discussing this in the properties of the nucleic acids. Okay, and the formation of the nucleosides. Okay, so this one we have the nitrogenous bases adenine, okay, over here, and this is getting linked to the pentose sugar by beta and glycosidic bond. This is the glycosidic bond, which is a covalent bond. Okay, so the 9th nitrogen atom is involved and the carbon 1 is involved. Okay, so this is called as the beta and glycosidic bond. Okay, since this is a beta form of ribose, so we call this as a beta and glycosidic bond. Okay, and the name for this nucleoside, adenine nucleoside, is adenosine. Okay, so guanine and adenine, they end with the O signs. Okay, adenosine and guanosine. Okay, their name end with the signs. Okay, while for the pyrimidines, their name ends with the signs only. So their name ends with the dines. Okay, so the pyrimidines, dine, D-I-N-E, and for the purines it will be the signs. Okay, so this is the major terminology given to the purines and pyrimidines. Okay, so adenosine contains the adenine and ribose sugar, while deoxyadenosine contains the adenine and deoxyribose sugar. Okay, the same name goes for the guanine, deoxyguanine, uridine, cytidine, deoxycytidine, or thymidine or deoxythymidine. Okay, so this is how the nucleosides are made. Okay, so this is the primary level structure for the formation of the nucleic acids. Okay, if we add one phosphoric acid group, Okay, so HPO4 minus. Okay, if we add a phosphoric acid group onto the fifth carbon position, that becomes a nucleotide. Okay, so nucleotide contains the adenine plus ribose sugar and a phosphoric acid group. Okay, so the numbers of the phosphoric acid will is different in NMP, NDP, and NTP. NMP nucleoside monophosphate contains one phosphoric acid group, while nucleoside diphosphate contains a two phosphoric acid, and NTP contains a three phosphoric acid groups. Okay, so the presence of phosphoric acid changes the names of these nucleosides. Okay, so now the name is called as adenylate acid or adenylate, or deoxyadenylate acid or the deoxyadenase, because the phosphoric acid is involved and the entire molecule got the negative charge on it. Okay, so the naming also changes. So the nucleotide names will be adenylate acid, guanylate acid, and citylic acid or iridylic acid if they are present in the RNA. So the same name is given in the DNA also. The deoxyadenylate acid, deoxyguanylate acid, because they contain the deoxyribose sugar. Okay, and the phosphoric acid. Okay, so nucleoside contains the base and sugar, while nucleotide contains the base, sugar, and also phosphoric acid groups. Okay, so this is the simple structure of the ATP, which contains an adenine molecule as a nitrogenous base and a ribose sugar as a pentose sugar, and three phosphate group as the uh, uh, three phosphate molecules. Okay, so which are linked to this ribose sugar. Okay, and if you talk about the bonds involved in the formation of this nucleotide, a big nucleotide. Okay, so the red one which you are seeing over here, this is an acid anhydride bond. Okay, so we have two of that. We have two of them. Okay, acid anhydride bonds, and the bond which is present between this 5 carbon and the phosphate group is known as the ester bond, and the bond which is present between the nitrogen atom and the first carbon of the pentose sugar is called as the glycosidic bond. A covalent bond. Okay, so this acid anhydride bond is actually a very energy rich bond. Okay, so and the simple molecule of ATP nearly gives about 7.6 kilocalories for all the calories of energy for all energy absorbing reactions. Okay, so the same amount of the energy is also it will also come from the GTP or UTP also, but mostly ATP will be acting as the energy donor in a lot of metabolic reactions. Okay, because it contains the acid anhydride bonds and the three phosphate groups in it produces much more energy. Okay, like 7.6 calories around. Okay, so since it has three phosphate groups, this is regarded as the ATP, so, or you can also call it as the NTP. ATP is nothing but the adenosine triphosphate, and NTP is also called as a nucleotide triphosphate. Okay, so nucleoside, it might be guanosine, adenosine, or cytosine, or anything. Okay, so this is a ATP, adenosine triphosphate. Okay, so if it contains only one phosphate, then we call that as the adenosine monophosphate. If it contains two phosphates, then we call them as then we call that as adenosine diphosphate. If it has three, so that will be the normal ADP. Okay, and if it doesn't have any kind of phosphoric acid groups, if it only contains only the pentose sugar, so the structure will be a simple adenosine, that is a nucleoside molecule. Okay, so this is about the nitrogenous bases, how the nitrogenous bases form the nucleosides, and how this nucleoside in turn get converted into the nucleotides. Okay, and the different bonds which are involved in the uh, nucleotides. Okay, so if you look at the phosphoric acid, we have the negative charge over an oxygen atom, so this gives the negative charge to the entire DNA. Okay, so that is why the DNA is negatively charged molecule, and this DNA being negatively charged molecule attracts the positively charged amino acid containing proteins. Okay, like histones, protamines, and all these proteins. Okay, so and also a lot of other drugs. Okay, which contain the positive charge. So that is why we can easily able to modify the structure of the nucleic acids inside the bacterial cell or in mammal cell. Okay, and one major property of this nucleotides is to exhibit the uh, is to absorb the UV light. Okay, so because these nucleotides has this uh, aromatic ring, the presence of the aromatic ring and the localization of the pi electrons in the aromatic ring, you know, helps them to absorb the UV light. Okay, so when you place a sample containing the nitrogenous bases in double beam UV visible spectrophotometer, and when you pass the light to that sample, so these nitrogenous bases, because they contain the aromatic rings, they absorb the UV light of 260 nanometers at different molar excitation coefficients. Okay, so the molar excitation coefficients for the adenosine is different, uh, and guanosine is different, and cytosine, pyrimidine, thymidine is also different. Okay, but they do absorb the light at around 260 nanometers. Okay, so this is the absorption spectra. uh, Highest molar accretion coefficient is seen for the AMP, adenosine monophosphate, and then the guanosine monophosphate. So why this adenosine monophosphate and guanosine monophosphate exhibit the highest molar accretion coefficient is they contain the double rings. Okay, one benzene ring and also one imidazole ring. Okay, because of presence of two rings, and the aromaticity also get increased, localization of the pi electrons also gets increased, and the molar accretion coefficient values also gets increased. Okay, so they quite absorb the wavelength at 260 nanometers. Okay, much highly. Okay, and then comes the pyrimidines. Okay, so not only these nucleotides, the aromatic amino acids, tryptophan, tyrosine, and phenylalanine, also absorb the UV light around 280 nanometers. Okay, the wavelength is different for the aromatic amino acids, and tryptophan uh, generally has highest molar extinction coefficient than the phenylalanine and then the tyrosine. Okay, so the molecule which contain the aromatic amino acids or the aromatic bases or aromatic rings in it, they tend to absorb the UV light. Okay, and the range is different for the aromatic amino acids and the purines and pyrimidines. Okay, and the chromophores which are present in the plants, they also absorb the. Okay, so the chlorophyll molecules and the phytochromes and all these things, xanthophylls and the carotenoids, they also absorb the UV light. Okay, so this helps in identifying the compound which is present in your sample. Okay, so if you are placing a sample which contains only nitrogenous bases, then you will see the peak absorption at the 260 nanometers. Okay, if your sample contains a lot of aromatic bases, if your sample contains the proteins, okay, so then the peak absorption will be at around the 280 nanometers. Okay, so this uh, absorption spectra will determine what compounds are present in your sample. Okay, when you are doing in normal laboratory things. Okay, so this is about the UV absorption peaks, the major property exhibited by the nucleotides because they contain the aromatic bases. Okay, if you talk about the functions of this individual NTPs, adenosine triphosphate or guanosine triphosphate or the cytosine triphosphate, any other NTPs. Okay, all these NTPs has different types of functions in the body. Okay, and majorly adenosine triphosphate, it acts as the storage battery in our tissues, and it actually releases the when it undergoes hydrolysis, it releases the 7.6 calories of energy for all the undergoing process. Okay, for all the energy absorbing process or the metabolic reactions which requires the ATP. Okay, example, synthetases are the reactions which utilizes the ATP molecules to synthesize some kind of molecules. Okay, by mixing the two different types of substrate and making the a bigger substrate. Okay, so those kind of synthetases reactions involve the ATP. Okay, and when you combine the citrulline with aspartate, okay, so we get arginosuccinate. Okay, okay, so this reaction is the second step in the urea cycle to form the arginosuccinate and then the ornithine and then the urea. Okay, so this step actually requires a special enzyme called as arginosuccinate synthesis. Okay, so this synthetase combines the citrulline and the aspartate to form the arginosuccinate in the urea cycle. Okay, for that it requires the ATP. Okay, So there are two types of synthetases, synthesis categories of categories. Okay, like the enzyme which synthesizes a molecule. Okay, is regarded in this category, synthesis category, and there are two different types of enzymes. Okay, so one is synthetase and one is synthase. Okay, so the major difference between the synthetase and synthesis. Synthetases utilize the ATP molecules, okay, while synthases they don't utilize the ATP molecules. Okay, so this is the major difference between the synthetases and synthesis. Okay, and apart from this, ATP is also required for the synthesis of the phosphocreatine from creatine. Okay, so uh, creatine is a molecule to store the phosphate groups. Okay, or excess phosphate in your body is stored in the form of phosphocreatine. Okay, so excess phosphate goes and binds to the creatine and forms the phosphocreatine. Okay, and whenever there is a requirement of phosphate for the phosphorylation of this ADP to ATP, so then this creatine donates the phosphate molecules. Okay, so ATP is also required for the synthesis of the phosphocreatine. So when there is excess ATP or when there is excess phosphate in the body, so phosphocreatine is synthesized. Okay, and this ATP is also required for the synthesis of the fatty acid from the acetyl CoA. So acetyl CoA is a precursor for the formation of the long chain fatty acids. Okay, and ATP is produced when you break down this fatty acids. Okay, this is exactly reversal for the synthesis. Okay, and this ATP is also required by the peptide synthesis or the protein synthesis. Okay, and proteins normally involves the GTP. Okay, and for formation of the glucose from pyruvic acid, okay, the reversal pathway, the gluconeogenesis also requires the ATPs. Okay, and for formation of the pyruvic acid also requires the ATP. Okay, and ATP is an important source for the muscle contraction, transmission of the nerve impulses. We all know this, okay, transportation of those molecules across the membranes. Okay, these active transport mechanisms utilize the ATP molecules. Okay, to transport the big molecules such as amino acids, sugars, and a lot of other big compounds in and out of the cell. Okay, to transport them in and out of the cell, they require the ATP molecules as an energy donor, and utilizing those ATP, they transport the molecules. Okay, and motility of the spermatozoa also utilizes a lot of ATP, and this ATP is provided by the mitochondria in the spermatozoa. And ATP is also required for the formation of the active methionine, which is required for the methylation process. Okay, so when we are methylating histone proteins, okay, the lysine amino acid in the histone proteins. Okay, so we require this active methionine. I'll tell you about the active methionine. And ATP is the major source for the phosphotransferase reaction. Okay, so that means whenever you want to add a phosphate molecules to the molecule, okay, you require a special type of enzymes known as the kinases. Okay, so these kinases adds phosphate molecule to any kind of molecules. Okay, so the major important kinase which we see in the glycolysis pathway is the hexokinase. So it transfers the phosphate molecule to the glucose, so and make this glucose into glucose 6 phosphate. Okay, and in vivo, inside the cell in the signal transduction pathways, this ATP is converted into cyclic AMP in the presence of adenylate cyclase enzyme. Okay, so this cyclase convert the ATP to cyclic AMP. Okay, so when it gets when the cell gets some sort of signals, like to break down the glycogen or to increase the growth factors in the cell. Okay, so at that time, this cell activates the cyclic AMP molecules. Okay, this cyclic AMP are responsible for the activation of the certain different protein kinases, and this protein kinases in turn activates a lot of other molecules inside the cell. Okay, so this ATP is converted into cyclic AMP inside the cell. Okay, as I said you, this creatine is the storage house for the phosphate molecules. Okay, when there is excess phosphate, that is stored in the form of phosphocreatine. Okay, and this phosphocreatine again donates the phosphate molecules to the ADP for the conversion of ADP to ATP. Okay, so when you add a inorganic phosphate to this ADP and in the presence of a phosphokinase enzyme, okay, it will convert into ATP molecule. Okay, and the same ATP cycle is also seen in the ADP synthase protein, which is present in the inner mitochondrial membrane. Okay, so this pump, ATP synthase pump, also convert the ADP to ATP by utilizing an inorganic phosphate molecule. Okay, in the inner mitochondrial membrane. Okay, so this phosphate not only involves in the conversion of the ADP to ATP, but it also involved in the formation. It is also involved in the muscle contraction, transmission of the nerve impulses, active transport, a lot of energy absorbing reactions, and the phosphorylation reactions. Okay, lot of reactions. Okay, and mainly this phosphate comes from the oxidative phosphorylation, or in the conversion of 1 comma 3 bis phosphoglycerate to 3 phosphoglycerate to 1 comma 3 bis phosphoglycerate, or when you break down the 1 comma 3 bis phosphoglycerate into phosphoenol pyruvate, you also get the phosphate. Okay, and from the succinyl CoA also, when you convert the succinyl CoA to succinate, you also get this phosphate molecules. Okay, so these substitutable reactions also donate the phosphate molecules. Okay, and this phosphate is also utilized by this kind of substrate level phosphorylation reactions. Okay, so this is about the phosphocreatine, and that is why the a lot of bodybuilders take this creatine molecule as to pump up their muscles. Okay, because it stores the phosphate molecules, and it involves in the muscle contraction. A lot of bodybuilders also take the creatine molecule, creatine powder. Okay, so that is a extra thing. And next nucleotide is the guanosine triphosphate, and the major role of the GTP is seen in the rhodopsin cycle. Okay, so this GTP actually activates a GMP phosphatidylase enzyme. Okay, and it inhibits the formation of the GMP, or it allows the conversion of the GMP to GTP, and this GTP blocks the sodium ion channels and the potassium ion channels in the vision cycle. So that gives you the vision. Okay, clear vision. Okay, so this GTP is mainly plays a important role in the vision cycle. Apart from it, this GTP is also in on the protein synthesis. Okay, so there are two elongation factor during the protein synthesis. So one is the EFTU and one is the EFTS. So this is the elongation factor thermo unstable, and this is the elongation factor thermo stable. This elongation factor thermo unstable utilizes the GTP molecule to translocate the amino acid onto the P side of the ribosome. Okay, so while the translation process is going on, the ribosomes, okay, so they require the amino acids on the P side, okay, to synthesize the long peptide chain. Okay, for this requirement, the EFTU utilizes one GTP molecules and takes the charged amino acid and places this charged amino acid onto the P side, or the peptidyl transferase activity side. Okay, and helps in the promote the peptide synthesis process. Okay, while the elongation factor TS, it regenerates the formed GTP into GTP. Okay, and again the GTP is utilized by the EFTU for the translocation process. Okay, so that is how the GTP is involved in the protein synthesis. Okay, raw protein synthesis. Okay, by the ribosomes. Okay, and GTP is also involved in the formation of the cyclic AMP in the G protein coupled receptor signaling pathway. Okay, so we have a big G protein over here, and to this G protein we have small G proteins, G alpha. Okay, and we also have a G beta, and we also have G gamma. Okay, which are linked to the plasma membrane. Okay, so when this big G protein gets signal, okay, so it in turn activates the G alpha protein. Okay, so this G alpha protein gets converted into active form G alpha GTP. Okay, okay, so G alpha GTP, and this G alpha GTP in turn activates the adenylate cyclase enzyme. Okay, which converts the ATP to AMP. So this is the initial signaling process in the G protein coupled receptor signal transfer pathway. Okay, so the GTP is necessary for the formation of the 3,5 cyclic AMP, and the GTP regulator side, the guanine nucleotide side, stimulation of adenylyl cyclase enzyme by hormone receptor complex requires the presence of GTP. Okay, like G alpha requires the GTP to stimulate the adenylyl cyclase enzyme for conversion of ATP to AMP. Okay, so one major role is in the signal transduction pathway, and I have said you in the rhodopsin cycle or the vision cycle, GTP is also required, and GTP is also required for the gluconeogenesis pathway, and it also required for the purine synthesis. Okay, so when we talk about the inosine monophosphate biosynthetic pathway, the synthesis of the inosine monophosphate, we will be seeing how this GTP is involved, okay, in the biosynthetic path, okay. And then we have the UTP. The major role of the uridine nucleotides is seen in the glycogen synthesis, okay. So in the glycogen biosynthesis pathway, this uridine UDP actually combines with the glucose 1 glucose 1 phosphate and forms UDP glucose, okay. So uridine diphosphoglucose. This uridine diphosphoglucose donates the glucose to glycogen primer, okay, like the primer which synthesizes the long chain of glycogen molecule, okay, that is called as the glycogenin, okay. It is actually a protein linked with the glucose molecule, okay. So this UDPG donates the glucose molecule to that primer, so that this primer gets elongated and entire glycogen molecule gets synthesized, okay. Polysaccharide gets synthesized, okay. And in order to form the UMP, in order to convert the UMP to UDP or UTP, so actually UMP is formed by the de novo biosynthetic pathway in the pyrimidine synthesis, okay. First is UMP is formed, and then later this UMP is converted into UDP or UTP by a phosphokinase enzyme. So the kinase enzyme which adds a phosphate molecule to the UMP, okay, makes it to UDP and UTP, okay. So this diphosphokinase enzyme which convert the UMP to UDP or UTP is called as a NUDKI, okay. NUDKI is an enzyme which is a kinase, which has a kinase properties, okay. In the presence of ATP, it converts the UMP to UDP and UTP, okay. And in the biosynthetic pathway of this UMP synthesis, the 5-phosphate orogenic acid decarboxylase undergoes the decarboxylation. Orogenic acid undergoes the decarboxylation in the presence of this decarboxylase enzyme and converts into UMP, okay. So I'll discuss the entire UMP synthesis in the next class, okay. As I've said you, so this UTP reacts with the glucose 1 phosphate to form the uridine diphosphoglucose, okay, in the presence of enzyme called as the UDPG phosphorylase. And this UDPG or UDP glucose, this can donate the glucose to a primer, okay. This primer is also called as the glycogenin, which is involved in the synthesis of this glycogen, okay. Glucose to a primer molecule of glycogen and is involved in the glycogen synthesis, okay. So this is how long polysaccharide gets synthesized in the liver, okay. And the major role is played by the UDPG to donate the glucose molecules, okay. So the formed UDPG, uridine diphosphoglucose, can also get interconverted or epimerized to form the UDP galactose, okay. So these are the epimers of each other, like glucose and galactose are the epimers, okay. So the special enzyme which converts the glucose to galactose is the epimerase, okay. UDP glucose galactose epimerase converts the UDP glucose to UDP galactose, okay. So these two molecules formed UDP glucose and the UDP galactose now gets oxidized to form the glucuronic acids, okay. UDP glucuronic acid and UDP galactronic acid, okay. So these are formed by the oxidation. So when this both UDPG and UDP gal oxidized, UDP glucuronic acid and the UDP galactronic acid are formed, okay. So this oxidized compounds, this formed glucuronic acid and the galactronic acid, they involved in the detoxification process and also in the conjugation process and also in the biosynthesis of this mucopolysaccharides of the glycosaminoglycans, okay. So formed UDP glucuronic acid mainly involves in the conjugation and the detoxification of bilirubin, benzoic acid, sterols, estrogens and the drugs, okay. So this is involved in the detoxification and conjugation process. Apart from conjugation and detoxification, this is also in the biosynthesis of this MPs and the GAGs, okay. So these are MPs are mucopolysaccharides, and you can also call this mucopolysaccharides as the glycosaminoglycans, okay. So these glycosaminoglycans or mucopolysaccharides are hyaluronic acid, heparin, chondroitin sulfate and all these things, okay. But formation of the chondroitin sulfate requires the UDP galactronic acid, okay. So this is the only difference between the glucuronic acid function and the galactronic acid function, okay. So for biosynthesis of this hyaluronic and heparin requires the UDP glucuronic acid, while biosynthesis of this chondroitin sulfate requires the UDP galactronic acid. This is a major important point you have to remember, okay. And then we have the cytidine nucleotides. Cytidine also important precursor for this cell wall polysaccharides. Okay, so actually cytidine monophosphate uraminic acid is involved in the formation of the cell wall polysaccharides inside the bacteria, and it is also involved in the formation biosynthesis of this phospholipids. Okay, CDP, cytidine diphosphate, forms or conjugates with the choline or glycerol or ethanolamine. Okay, and they are involved in the formation of the phospholipids. Okay, or actually biosynthesis of the phospholipids. Okay, and when cytidine monophosphate combines with an oligosaccharide called as the sialic acid, okay, it helps in the biosynthesis of the salivary mucin. Okay, so the salivary mucin actually secrete from the salivary glands of salivary glands. Okay, and for this biosynthetic pathway, it actually requires the CMP sialic acid. Okay, and major role is seen in uh, biosynthesis of the phospholipids. Apart from the biosynthesis of the phospholipids, it is also involved in the formation of the polysaccharides, and it is also involved in the formation of the salivary mucin. Okay, and then what we have is a PAPs. Okay, so I've said you about the active sulfate molecule, so which is formed from ATP. So this active sulfate will will be involving in the transfer of sulfate groups to various substances or various biomolecules. Okay, like for the heparin or the chondroitin sulfate. Okay, so these mucopolysaccharides also require the sulfate molecules. Okay, for their activities. So to get the sulfate molecule, they utilize the PAPs, phosphoadenosine phosphosulfate. Okay, which is found from the ATP. Transfer the sulfate molecule to the heparin, chondroitin sulfate, or keratosulfate. Okay, all these things. Okay, and there are also a lot of sulfolipids. Okay, uh, for the formation of all these compounds, PABs donate the sulfate molecule to these compounds. Okay, and helps in the biosynthesis. Okay, for formation of the sulfolipids or sulfates. Okay, so it is also required, and it is also required for the conjugation of phenols, indols, catol to form the etheryl sulfate. Okay, so actually conjugating a sulfate molecule to the compounds. Okay, PABs is required, and cyclic AMP, which is formed from the ATP, will act as a secondary messenger. Okay, so major role of the cyclic AMP is to act as a secondary messenger and to amplify the initial signal into 10,000 times greater. Okay, 10,000 or 1 lakh times greater. Okay, so it also regulates the glycogen metabolism when there is a glucose level drop down in blood. It stimulates the glycogen metabolism, like to glycogenolysis pathway, to break down the glycogen into glucose molecules. Okay, so that is actually done by the cyclic AMP molecule, which is a secondary messenger. Okay, so when there is increased level of cyclic AMP inside the liver, okay, so that stimulate the glycogenolysis and that break down the glycogen to small glucose molecules. Okay, it also regulates the TG metabolism to break down the triglycerides. Okay, and it also inhibit the cholesterol biosynthesis. Okay, this is the major role of this cyclic AMP. Okay, it is all during the starvation conditions, it also inhibit the cholesterol biosynthesis. Okay, only in the starvation conditions. Okay, and this also cyclic AMP, the major function of the cyclic AMP is to activate the protein kinases. Okay, so inside the cell, whenever there is a signal, whenever and there is a rise in cyclic AMP levels, this cyclic AMP goes and stimulate the protein kinases and makes the protein kinase into active form. Okay, and then the protein kinase further activates and different kind of proteins or compounds inside the cell. Okay, and the cyclic AMP also modulates the transcription translation and the protein biosynthesis pathways. Okay, so these are like different functions of the cyclic AMP. There are a lot of important functions. Okay, it also regulates the insulin secretion and melatonin synthesis and the catecholamine biosynthesis. Okay, and then we have the cyclic GMP, the GMP which is involved in the rhodopsin cycle. So when there is cyclic GMP, the vision will be dark. When there is GTP, the vision will be clear. Okay, so that is during the daytime. Okay, so in the dark times, okay, or in the night times. Okay, light dark adaptation, in the light dark adaptation cycle. Okay, so in the night times, cyclic GMP is produced, and this cyclic GMP allows the sodium and potassium ion channels to influx or efflux the ions. Okay, so this results in the loss of vision. Okay, loss of vision in the sense for the temporary time. Okay, so this GMP plays a major role in the retinal light dark adaptations. Okay, and GMP also involves in vasodilating the smooth muscle cells. Okay, relaxation of the smooth muscle cells is done by the CGMP, and the level of CGMP is raised by certain type of compounds called as a nitroglycerin or the sodium nitrate. Okay, so this nitroglycerin and sodium nitrate actually increases the cGMP levels, okay, around the smooth muscles, okay, and that causes the smooth muscle to relax. Okay, so the major role of the cyclic GMP is to relax the smooth muscles or to vasodilate the smooth muscles, or to involve in the rhodopsin cycle, and also regulate the insulin actions, okay, and also regulate the neurotransmitter efficacy. Okay, so how much amount of the neurotransmitter has to released in the synaptic terminals, okay, synaptic in the axon terminals, okay, so that efficacy is controlled by the cyclic GMP. Okay, so these are major roles of the cyclic AMP and cyclic GMP. Okay, and in the next class we'll be discussing about the biosynthesis of the nucleotides. Okay, uh, how the nucleotides are formed and the different pathways involving in the formation of the nucleotides. Okay.